Welcome to Lecture 4. This lecture is on protein composition and structure. It follows Chapter 4 of your textbook, sections 4.1 to 4.4. I've titled this lecture, What is a Protein? So let's get started. To give us a good primer, please watch this video from the Research Collaboratory for Structural Bioinformatics, RCSB, for a nice primer background on proteins.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas on the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB Protein Databank.
Now that you've watched that video and seen the diversity of proteins and their functions, I want to talk about the human proteome. So the human proteome was once thought to be on the basis of a one gene to one protein type of function. So it was predicted that perhaps 20,000 proteins were in the human proteome. Now we know there are actually many other things happening, including alternative splicing events of the transcribed genes and post-translational modifications of proteins. So instead of one gene to one protein, we're getting one gene to many proteins from the human genome into the human proteome. Proteins are composed of amino acids, and amino acids are shown in this picture. The carboxy terminus of the amino acid is linked to the next amino acid through a peptide bond, which is formed by the condensation reaction. You can see here that the condensation reaction, as the name implies, a water leaves from the formation of this peptide bond. Through the condensation reaction, you can form a peptide, a dipeptide made of 2 amino acids, a tripeptide made up of 3 amino acids, and so on. A polypeptide, as we define it for our class, is going to be greater than 50 amino acids. And then a protein is made up of 1 or more polypeptide chains. So the ribosome is the holoenzyme complex that is translating your genetic code into proteins. Once the protein is translated, there is protein folding that is happening to create the next levels of protein structure, of which there are 4. So let's talk about protein structure levels. The 4 levels of protein structure are outlined in figure 4.3. First, we have primary structure, which is just the sequence of amino acid residues, as you see in the example. Secondary structure is how these residues come together in space. And another way to think of that is the orientation or conformation of the polypeptide backbone. Tertiary structure describes how those elements of secondary structure come together to create the three-dimensional structure of the polypeptide. And finally, quaternary structure, the fourth level of protein structure, is how these polypeptides come together and interact. Now let's review the textbook readings. What kind of information can be learned from a protein's primary structure? As I mentioned, the protein's primary structure is the amino acid sequence of the polypeptides that make up the protein. Based on the amino acid composition, which can be hydrophobic, polar, charged, you can determine the properties such as hydrophobicity and total charge at a certain pH, and you can also calculate the molecular weight of the protein. For more information, you can refer to figure 4.2. Now let's draw out this short peptide. This peptide is glycine, serine, tyrosine, cysteine, alanine, proline. Please pause and give it a try. I hope you tried drawing this peptide. Now you can see what it should look like. We have the amino terminus on the left-hand side, and you have the carboxy terminus on the right-hand side. You can see the peptide bonds that connect amino acids together. You will notice that for the residue side chains, for glycine, I haven't drawn another bond above the alpha carbon because the residue side chain is a hydrogen. You can see that for serine, I've drawn in the hydroxyl group for the side chain, and so on for the tyrosine side chain. I've drawn the hexagon shape with a ring in the middle to denote the aromatic nature of this residue. Cysteine, instead of the oxygen that is in the serine, has a sulfur atom, so SH. And lastly, you'll see the alanine followed by the proline. Be careful with the proline because in proline, the amino end of the residue is connected to the alpha carbon. Secondary structure describes the shape of the polypeptide backbone. Can you describe three major classes of secondary structure? The three major classes of secondary structure are alpha helix, as you can see here, beta strand, which describes one of these strands. When you have two of them or more, you can start to call it a beta sheet. And you can say that the beta strands are parallel if they are forming in the same direction, or anti-parallel if they point in opposite directions. Lastly, you have irregular or disordered type of secondary structure, which just generally means that the backbone does not have a regular structure. You find these in instances when the protein is coming together into its folded form. So for example, this alpha helix is connecting to this beta strand through an irregular disordered region, and the beta strand is then connecting to the next beta strand through another irregular disordered region. The next set of questions is on tertiary structure. Pause here and answer these questions. Great. What is the major determinant of tertiary structure and protein folding? Well, because proteins are synthesized in an aqueous environment in the cell, the major determinant of tertiary structure is how hydrophobic the protein is. Hydrophobic regions of the protein cannot make hydrogen bonds with water, which is polar, and so it will arrange itself in a way to avoid interaction with water. We call that the hydrophobic core. What kind of amino acids will you find on the surface of a protein as opposed to in the core? So when a protein is folded, you can expect to find the hydrophilic amino acids on the surface interacting with polar water, and hydrophobic amino acids forming the core, as I said, the hydrophobic core of the protein. Now I'd like you to give 3 examples of cross-links that help stabilize the folded state of proteins. Three examples of cross-links that help stabilize the folded state of proteins. First, we have the ionic bond, and I've pointed out the aspartate residue here, interacting with the arginine residue here from 2 different sides of a protein. And because they have opposite charges, they're able to interact. Next, we have a disulfide bond, which is a covalent bond between one cysteine residue and a second cysteine residue. As you might recall, the cysteine residue has a sulfhydryl side group. They each lose that proton and form this covalent bond. Lastly, with an inorganic metal ion, you can have different parts of a protein coordinate that metal ion. In this case, I'm showing a zinc finger motif. So the zinc ion is being coordinated by these cysteine residues that I've drawn here. And so the zinc ion is actually acting to stabilize this orientation of the protein. Lastly, quaternary structure is when multiple polypeptides that each have their own tertiary structure come together to form the functional protein. And as mentioned before, there are cases where proteins consist of only one polypeptide. Let's answer these questions. The difference between a heterooligomer and a homooligomer is that a hetero implies different proteins coming together, and homo should imply to you the same proteins are coming together. In the example of hemoglobin, 2 alpha subunits are combining with 2 beta subunits. That's a total of 4 subunits. But since there's at least 2 different types of subunits, we'll call it hetero. And because there are 4 of those subunits, we'll call it a tetramer. So the answer is a heterotetramer. Here's our last exercise for this lecture. Go ahead and list the forces that can hold quaternary structure together and chemicals and methods that can denature a protein's folded state. Similar to interactions that hold tertiary structure together, the forces that can hold quaternary structure together are hydrophobic interactions, hydrogen bonds, ion pairs, and lastly disulfide bonds. If we aim to disrupt quaternary and tertiary structure, the forces, for example, listed above, we can use different chemicals and methods to overcome them. So for example, you can heat or boil a sample to 95 degrees Celsius, and that will break up a lot of bonds. Reducing agents, such as dithiothiritol, DTT, beta-mercaptoethanol, BME can reduce disulfide bonds. Lastly, with salts, when you have large amounts of ions, they can disrupt ionic pair bondings. Now that we've talked about the 4 levels of protein structure and how they scaffold on top of each other, we can categorize proteins into 3 protein classes. First, we have globular, which contains defined hydrophilic surface and hydrophobic core. They're usually composed of 2 types of secondary structure. Examples are the hemoglobin subunits, trypsin, amylase. Second protein class we have here is fibrous. It often functions as structural material by acting as fibers and filaments in and outside of the cell. They can be composed of globular domains or subunits. Examples here are keratin, collagen, filamentous actin. Lastly, we have membrane proteins. Now, if we have a lipid bilayer membrane, a protein that spans the membrane, meaning it's present on both sides of the membrane, is called an integral membrane protein, whereas if it just associates with the membrane nearby, we call it peripheral. And often they are peripheral via hydrophobic surface interactions on the surface of the protein, or through lipid-linked anchoring mechanisms. So if a hydrophobic group was put on that protein, it could be anchored into the membrane. And examples of membrane proteins include receptor proteins, channel proteins in the cell membrane. That wraps up our lesson on protein structure and function. I've left you some extra material that you will not be tested on, but it is enriching. Now that you know more about peptide bonds and proteins, you can learn about the protein folding paradox and how protein misfolding and unfolding applies to the real world in terms of human disease. Thanks, and I'll see you in class.